Hello everyone. My name is Anushanand, working as a tutor in M College of Nursing. In this video, we will talk about chemotherapy. In this video, we will discuss about definition of chemotherapy, objectives, goal of chemotherapy, cell cycle, how does chemotherapy kill cancer cell, classification of chemotherapeutic agents. Chemotherapy is the use of drugs to destroy cancer cells. It usually works by keeping the cancer cells from growing, dividing, and making more cells. Because cancer cells usually grow and divide faster than normal cells, chemotherapy has more of an effect on cancer cells. Definition of chemotherapy. Chemotherapy is the use of anti-neoplastic drugs to promote tumor cell destruction by interfering with cellular function and reproduction. It includes the use of various chemotherapeutic agents and hormones. Objectives of chemotherapy. Chemotherapy maximizes the death of malignant tumor cells. It will cure the client with cancer. It controls the tumor growth when cure is not possible. It extends the lifespan and improves the quality of life of client with cancer. Goal of chemotherapy. The goal of chemotherapy depends on the type of cancer and how far it has spread. Sometimes the goal of treatment is to get rid of all the cancer and keep it from coming back. That is curative. If this is not possible, you might receive chemotherapy to delay or slow cancer growth. Delaying or slowing cancer growth with chemotherapy also help manage symptoms caused by the cancer. Chemotherapy given with the goal of delaying cancer growth is sometimes called palliative chemotherapy. Cell cycle. The cell cycle is an ordered series of events involving cell growth and cell division that produce two new daughter cells. Cells on the path to cell division proceed through a series of precisely timed and carefully regulated stages of growth, DNA replication, and division that produces two identical cells. The cell cycle has two major phases, interphase and the mitotic phase. Interphase is again divided into three stages, G1, S, and G2 phase. During interphase, the cell grows and DNA is replicated. During the mitotic phase, the replicated DNA and cytoplasmic content are separated and the cell divides. How does chemotherapy kill cancer cells? Chemotherapy damages the cells as they divide. In the center of each living cell is the nucleus. This is the control center of the cell. It contains chromosomes, which are made up of genes. These genes have to be copied exactly each time a cell divides into two to make new cells. Chemotherapy damages the gene inside the nucleus of cells. Some drugs damage cells at the point of splitting. Some damage the cells while they are making copies of all their genes before they split. Chemotherapy is much less likely to damage cells that are at rest, such as most normal cells. A combination of chemotherapy drug is often used. This will include drugs that damage cells at different stages in the process of cell division. Some chemotherapy medicines can kill a cell during any phase of the cell cycle. They are called cell cycle nonspecific agents. Other chemotherapy medicines kill cancer cells only during a certain phase. They are also not able to work in the resting phase. They are called cell cycle specific agents. Let's move on to the classification of chemotherapeutic drugs. Cancer chemotherapy agents are classified according to their chemical nature and function. Some of the well-known classes of cancer chemotherapy agents are alkylating agents, antimetabolites, anthracyclines, plant alkaloids, topoisomerase inhibitor. Now we will deal one by one. Alkylating agents. Alkylating agents are the oldest and most commonly used class of chemotherapy drugs and work by directly damaging DNA and preventing cancer cells from reproducing. They are used to treat a wide variety of cancers but have the greatest effect on those that are slow growing. They are cell cycle phase nonspecific, meaning that they kill cancer cells in any phase of the cell cycle. Some examples of alkylating agents are shown: mustard gas with derivatives, ethylenamines, alkaline sulfonates, hydrazines and triazines. Antimetabolites. Antimetabolites are chemotherapy drugs that interfere with DNA and RNA growth. They are cell cycle specific, meaning that they kill cancer cells in a specific phase of cell division. Some examples of antimetabolites are folic acid antagonist, pyrimidine antagonist, purine antagonist, adenosine deaminase inhibitor. Anthracyclines. Anthracyclines are anti-tumor antibiotics that interfere with enzymes necessary for DNA replication. They are cell cycle non-specific and are used to treat a variety of cancers. Some examples of anthracyclines are doxorubicin, mitomycin, etc. Plant alkaloids. The name itself suggests it derived from plants. Plant alkaloids are derived from certain type of plants found in nature and inhibit or prevent mitosis or inhibit enzymes from making proteins necessary for cell reproduction. Most plant alkaloids are cell cycle specific but can cause damage in all phases. Some examples of plant alkaloids are vincristine, vinblastine, etc. Topoisomerase inhibitor. Topoisomerase inhibitor act by weakening the structure of a cancer cell that is necessary for it to divide. There are other types of chemotherapy medications which are unique in their ability to attack cancer cells. Examples are topoisomerase 1 inhibitor and topoisomerase 2 inhibitor. Now I would like to conclude this video. In this video, we dealt with definition, objective, goal, cell cycle, how does chemotherapy kill cancer cell, and classification of chemotherapeutic agents. Hope you understood this topic. See you soon with next topic. Thank you.